Hello there. Today I am going to teach you about the hematopoiesis. This series of lectures of hematopoiesis will be completed in 3 lectures. So this is the part 1 in which I am going to teach you about the basics of hematopoiesis. I am going to talk about the blood, its component of the blood, and then the process of hematopoiesis in general. The name blood do come in our mind, immediately it comes that it is something red in color, moves in our body. So this blood is a connective tissue suspended in plasma, and if we quantify the blood, it ranges from 4 liter to 5 liter in a person, but it roughly about 8% of total body weight of an adult human of 70 kg weight is around 8% of body weight, which constitutes the blood cells, and these blood cells are suspended in what? Plasma. So these blood cells are also called as hematocrit or packed cell volume, which mainly includes the cellular part, that is red blood corpuscles, white blood corpuscles, and the red blood cells, red blood corpuscles, white blood corpuscles, and the platelets. Thereafter comes the plasma. Plasma is a water liquid component in which water, electrolytes, proteins, and nutrients, waste material are dissolved. So this total hematocrit or the cellular portion in total blood comprises around 45%. This 45% is a average, but in the male and female these are different slightly. In the male it's around 46%, and in case of females it's around 44%. This slight difference because females of reproductive age group have monthly menstrual cycle in which they do lose around 50 ml to 100 ml blood every monthly cycle. That is why their RBC are being lost, and that is why hematocrit is less. Out of this total hematocrit of this 45%, 99% of the hematocrit of this 45% is made up by the RBC. Rest 1% is contributed by the platelets, and this plasma is 55%. In an adult human, total plasma in a liquid form is around 3 to 3.5 liters, and it mainly constitutes of the albumin protein, globulin protein, and fibrinogen protein, apart from the nutrients and dissolved electrolytes, waste material. So if we take out the blood from the body and put into a test tube and spin it, we would see 3 different layers distinct from each other. Upper translucent light yellowish portion is known as plasma. It do not have any kind of the cell. The lower portion, this dark red, is made up of RBC or erythrocytes of 45%, and about 1% of the intermediate buffy coat is made up by the platelets and leukocytes. If you see under the microscope, we would see these are the red blood cells, these are the white blood cells or leukocytes, different sizes, and these are the platelets. Let's see, these blood cell contain what kind of cells? They contain the red blood corpuscles, white blood corpuscles or leukocyte. So these leukocyte are also, leuco means white, cyte means cell. So leukocytes or white blood cells can be subcategorized into two major categories, granulocytes and agranulocyte. Granulocyte means their cytoplasm do contain a specific protein when we do stain them, and these are the three different categories of granulocytes: agranulocytes, lymphocytes and monocytes. And there are third type of particle, that is platelets or thrombocytes. Let's see what they look like under the microscope. So these are the erythrocytes, red in color because they do have hemoglobin, and this heme when combines with the oxygen becomes red in color. That is why they look like red. Rest of the cells in the body are colorless until unless they have a specific color. So to look those cells under the microscope, when we do observe, we need to put some certain color so that we can differentiate their cell membrane, their nucleus and other structures. So this neutrophil, which a granulocyte, is characterized by multi-lobed nucleus. You can see it may have 3 different lobes of nucleus or 4 lobes, but large quantities of granules in the cytoplasm. Similarly, eosinophils have this bilobed, bi means 2, lobe means lobe, and have the granules. So they have acidic dye. Another one is this basic dye-stained cell, that is basophil. They do also have the bilobed nucleus, but the nucleus of the eosinophil and basophil difference is that they have H-shaped nucleus. Similarly, lymphocytes and monocytes do not have any granules, but large nucleus is there, and they are larger than lymphocytes, and these are the platelets. Let's see what are the functions of each constituents of the blood. So first we will look into the plasma. Plasma is constituted of these non-cellular molecules: water, electrolytes, nutrients, waste material, gases, especially oxygen and carbon dioxide, hormones and plasma protein. And there are cellular component. These cellular components: erythrocytes, leukocytes and platelets. So erythrocytes' main function is what? Transporting oxygen and carbon dioxide. 98.5% of the oxygen is transported by the hemoglobin, and around 23% is transported by the carbon dioxide. Hemoglobin is transporting the carbon dioxide. Amongst the leukocyte, neutrophil acting as a phagocytic cells. Eosinophils attacks the parasitic worms of Helminth group, and basophils releases histamine, which also creates the allergic reactions in the body. Monocytes remain in the blood circulation for 2-3 days, then they do migrate to the tissues. There it becomes the macrophage. Their size increases almost double. Lymphocytes are of mainly 2 types: B lymphocytes and T lymphocytes. B lymphocytes are also called B equivalent lymphocytes or Bursa equivalent. This first time these kind of lymphocytes were observed in birds in a specific anatomical location, that is called as Bursa febrigus. So that same name has been given to other type of cells which were observed in all vertebrate group. So B equivalent lymphocytes or simply B lymphocytes. So its function is to produce the antibodies against the immunity. The vaccines are also being produced by producing specific antibodies or memory B cells. And these T lymphocytes are of 3 types: regulatory T cells, helper T cells and cytotoxic T cells, and these T cells are the link between the innate immunity and the acquired immunity, that means amongst the B lymphocytes and the macrophages. And platelets, obviously it stops the blood loss from the blood vessel injury. So total process of this blood cell formation is known as hematopoiesis. So there are three different kind of cells: RBCs, WBCs and platelets, and their specific lineages. So formation of RBCs are called as erythropoiesis. Formation of white blood cells or WBC called as leukopoiesis, and it happens with two major lineages. One is myeloid lineages, another is lymphoid lineages. Lymphoid lineages creates the T lymphocytes and B lymphocytes, and this thrombopoiesis produces the platelets. So this hematopoiesis starts at the early stage of life within the placenta. In human embryo, this blood formation, the RBC formation, starts at the embryonic stage of 20 days by the blood islands present in the yolk sac. So these yolk sac have a specific kind of cell, is known as hemangioblast cell. Then this hemangioblast cell migrates to the heart vessels and then placenta and finally to the liver by the 30 days of embryonic life. When it reaches to the 70 days life, bone marrow is being formed, and this bone marrow starts producing this RBC. So this schematic diagram is just showing that how this RBC getting formation, hematopoiesis, starts at embryonic stage. So this is the yolk sac, these are the blood islands of these red cells, hemangioblast cells, and they are the mature RBC in the embryonic stage. And when it reaches to the 30 days embryonic life, liver start producing these red blood cells, and at the bone marrow 70 days, these are the all nucleated red blood cells, and the characteristics is different than the adult RBC. Adult RBCs do not have the nucleus, but these embryonic RBCs do have the nucleuses, and the hemoglobin have a different globin protein. Here is that eta and gamma type, alpha and epsilon type, eta and epsilon type. This 2 number means 2 polypeptide chain of the same protein, and when it grows in bone marrow, it changes from eta and epsilon to the alpha and beta type or alpha and delta type. Let's see. Here we do see the stem cells. Stem cells are the cell renewable cell which can live for the life and keep on dividing if they remain in a microenvironment of the bone marrow. So these hematopoietic stem cell, upon receiving a specific signal from the cytokines like this interleukin or colony stimulating factors or stem cell factors of a specific type like granulocyte or macrophage colony stimulating factors, it divides into particular lineages and creates its own offspring, that is the final cell. So it can be understood through an example of our day-to-day family life, like our great grandfather had many children, and those children, upon living in a specific condition, they had started creating their own family lineages. So each family lineage is specialized to produce specific cells. Here in case this GMCSF lineages produces the RBC through a series of development. Similarly, series of development creates the platelets, monocytes, neutrophil, eosinophil, basophils, or the lymphocyte. In another example of a society, we do see the doctor, engineer, lawyer, artist, helpers, drivers. They're doing certain specific profession, but all they are human. So since they are lived in a specific condition or environment which help them to become a adult human, so they choose a profession. Similarly, these are the environmental factor for these stem cell to stimulate certain cell into certain direction. So in this lecture, these lineages of RBC formation is known as erythropoiesis, and the formation of platelets is known as thrombopoiesis, and formation of monocyte, neutrophil, basophil, and eosinophil is known as myeloid lineages, and the lymphoid lineages is formation of these B cells and